Welcome to Beyond Biology Lab YouTube channel. Today we're exploring a fascinating space right at the intersection of synthetic materials and natural biology. We'll be looking at how emerging research is allowing us to bridge these two very distinct worlds to successfully regenerate human tissue. It's a deeply hopeful look at the steady, deliberate progress happening right now in regenerative medicine, drug discovery, and translational science.
Here's the step-by-step roadmap for our explainer today. First, we'll look at the tissue engineering challenge. Then, we'll move into engineering the perfect scaffold, the unique power of nanofibers, and the precise mechanics of magnetic cell seeding, before wrapping up with what this all means for the future of regeneration. Let's dive in.
Section 1. The Tissue Engineering Challenge.
One of the greatest hurdles we face in translational medicine today is our limited ability to grow complex, functional, three-dimensional tissues, like replacement blood vessels. To understand the solution to this, we first need to define a foundational tool called a scaffold. You can think of a scaffold as a structural biomaterial framework, much like the steel beams of a building. It provides a stable, comfortable environment for autologous human cells, the living inhabitants if you will, to anchor, multiply, and eventually regenerate their native extracellular matrix. These cells absolutely need this perfect physical environment to thrive. This brings us to a really crucial question driving the field. How exactly do we construct advanced replacement blood vessels that the human body will successfully integrate and accept? This is a highly specific, real-world clinical challenge, and it requires a remarkably precise approach to the biomaterials we use.
Section 2. Engineering the perfect scaffold.
Solving this biological puzzle begins by bringing together two very distinct materials to create an optimal growth environment. Right now, researchers are focusing on blending synthetic PCL, or polycaprolactone, with natural silk fibroin. PCL is an established FDA-approved synthetic polymer known for its high mechanical strength and its slow, controlled degradation. Silk fibroin, on the other hand, is a natural protein derived from the silkworm, which offers exceptional biocompatibility and naturally encourages human cells to attach to it. By carefully blending these two together, we get the best of both worlds: the mechanical backbone of synthetic PCL and the natural biological functionality of silk. Traditionally, mixing synthetic and natural polymers meant using harsh, toxic organic chemicals, but researchers are now using a clever, eco-friendly electrospinning approach with benign solvents, specifically a mixture of acetic acid and formic acid. Not only does this massively improve safety for lab workers and help with waste management, but crucially, it prevents the delicate silk proteins from denaturing. It preserves that vital biological integrity so the cells can actually use it.
Section 3. The Power of Nanofibers.
Now let's transition from the chemistry to the actual microscopic, three-dimensional physical architecture of the scaffold itself. When we look at this under a scanning electron microscope, there is a stark contrast in the microarchitecture. Standard PCL tends to create thicker microfibers, but when you blend PCL with silk fibroin, using those benign solvents we just talked about, it creates a much finer, tighter architecture made of submicron nanofibers. This highly porous, interwoven web is exactly what you want because it closely mimics the natural human tissue environments where these cells prefer to live. And we can actually measure how much the cells prefer it. If we look at metabolic activity, which is basically a marker for cellular health and reproduction, at day 7, the evidence is super compelling. Cells show baseline survival on standard microfibers, and they do a bit better on synthetic nanofibers, but they absolutely thrive on the blended PCL and silk fibroin. This is a vital distinction because these are living human endothelial cells and fibroblasts, the exact cells you need to structurally line and maintain a healthy replacement blood vessel. We can confirm this physically with fluorescence microscopy. By highlighting the cell cytoskeleton in red and the nuclei in green, we can clearly see highly optimized rapid cellular growth. After just 7 days, dense, thriving multi-layers of cells have successfully colonized that PCL and silk fibroin blend. It's clear proof that this hybrid material genuinely supports a robust biological foundation.
Section 4. Magnetic Cell Seeding.
Getting cells to grow on a flat petri dish is one thing, but efficiently coating the complex, three-dimensional inner surface of a tiny tube for a vascular replacement, that is an entirely different mechanical challenge. To solve this, researchers use a precise three-step process called 3D radial magnetic seeding. First, they load autologous human cells with harmless superparamagnetic iron oxide nanoparticles, often called SP-ions. Second, these magnetically loaded cells are carefully transferred into the hollow space, the lumen of the tubular scaffold.
Finally, they apply a radial magnetic field from the outside. This magnetic force gently and evenly pulls the cells directly against the inner walls of the tube. This ensures a perfectly uniform distribution, rather than just letting the cells settle at the bottom due to gravity.
This highly precise scientific process yields some incredible physical results. When you look at a macroscopic 6mm tubular scaffold and its microscopic cross-section, you can really see the success of this magnetic seating in action. Seating endothelial cells alongside supporting fibroblasts creates a highly uniform, natural biological lining. This tight, cobblestone-like monolayer provides the essential barrier required for real, functional vascular tissue engineering.
Section 5. The future of regeneration.
By synthesizing these laboratory findings, we can start to see a very clear, intellectually grounded path for the future of translational medicine. On a practical, clinical level, optimizing these scaffold materials and seating techniques directly accelerates the pipeline for personalized therapies using a patient's very own cells. It sets a solid foundation for faster, more reliable, and ultimately cheaper precision medicine pipelines, aiding everything from new drug discovery models to full tissue replacement. But as these theoretical models become clinical realities, it prompts some really vital conversations. We have to start asking new ethical questions regarding fairness and access, transparency in biomanufacturing, and the complex legal ownership of these newly engineered biological tissues.
As we continue to observe this incredible convergence of synthetic science materials and natural cellular biology, it leaves us with a profound question to think about. What does biology become next when we learn to engineer living systems with the exact precision of a synthetic circuit? It's a deeply hopeful yet complex horizon for science.
Thanks for watching. Please like and subscribe Beyond Biology Lab YouTube channel.